Are we done? What happened? Huh? Doubt है. हाँ बोलो.
They have given right.
कौन सा? अरे. Right answer wrong दे दिया. ऐसे कैसे बोला? One by one. अरे we love you so much. We all love you. अरे मुझे बैठना है कि सामने बैठना है? अरे इधर आ तुम्हारा दोस्त आ हम बोलो डांस फटाफट इतनी ना no read out
I mean, last class you were told us that you are more flexible than RBC because the doctors will have. But how is it possible? Because the RBC nucleus organelles, but WBC have nucleus.
I don't want any noise in the class. Number one. A 10th standard classroom should be very, very quiet. Each one of you know that he is asking a question. It's not only his doubt. I'm clarifying the doubt for all of you. So all of you are supposed to listen to this. ऐसा नहीं कि उसने पूछा तो उसको ही सुनने दो. Not that way. Now, what you ask is that if it is a RBC cell, the RBC cell does not have nucleus. WBCs have got big nucleus, and how come they are flexible? How come they are coming out of a capillary wall, capillary pore? This is very clear that when we talk about the RBCs, number one, the RBCs are small in size. They don't have nucleus. On the other hand, the WBCs are much bigger, and they have got big lobed nucleus. But again, that's also true that these WBCs are the ones which can change their shapes and come out through the very narrow capillary pores when the RBCs get stuck. It is only because of the special feature that the WBCs have, known as the amoeboid nature of the WBC, which can change shapes from different, according to different situations. That's how these WBCs can reach the site of injury also and bring about either the process of phagocytosis or release of antibodies, only because they can come out of the general circulation and reach the tissues. But the RBCs need not also, and they cannot also. Because the RBCs are very stiff cells. They don't have many other different type of cell organelles. And on top of that, it is not to understand this way that since they have, they don't have endoplasmic reticulum and nucleus, and they should have been more flexible. Not that way. Whatever shape they have, that is fixed. WBC, even though they are much bigger than the RBCs, they can change their shape. They can become narrow thread-like structure and skip through those pores. Amoeboid nature. The WBCs are always amoeboid in nature.
Are all leukocytes like that?
Mostly all. Mostly all. When we talk about the 5 types, if you remember, I showed you the other day. The neutrophils also can do that. The lymphocytes also can. Monocytes also can. Mostly all are amoeboid in nature. But majorly we understand the ones which are phagocytic, something like neutrophils, monocytes, they're massively they are amoeboid. आया समझ में? बोलो. तो पढ़ो ना. Read. क्या है क्वेश्चन क्या है? तुमने क्या आंसर लिखा था?
Mention 3 structural features of a chlorine which adds to the absorptive capacity.
What do you wrote?
Chlorine and Dct Membrane.
Mention 3 structural features of the small intestine, which adds to the absorptive capacity. और reliance अभी click करो ना.
अरे वो बोल रहा था आपने homework दिया है.
Now, what I said, what you have written is wrong. You have written the 3 structural parts of the small intestine, but the question is not that. The question is asking you, write the 3 structural features of the small intestine जो absorption में help करता है. And आपने जो लिखा, you had written 3 structural parts of the small intestine. वो तो question ही नहीं है. That's why I do a lot of questions. You should understand what the question is. आया समझ में? So what is the answer they have given in the volume in the model answer? You have to write about the villi, the wall of the villi, all these features, 3 structural features.
But beside that, we have to write the narrow and long
structural feature. वैसे तो 4-5 features है. If you have to write 3, one feature you will write. अरे, why was that big broad smile? Huh?
Ma'am, in the paper I wrote because the question was.
Let me finish this. So, what are the 3 structural features that help in the process of absorption? You're supposed to write. एक होगा the extensive coiled nature of the small intestine, ताकि the food will pass slowly and the process of absorption will happen. Number 2, the presence of numerous finger-like projections known as villi in the inner wall of the small intestine. अगर ileum याद है तो ileum. Third क्या लिखोगे? The large number of villi increase the surface area. Fourth feature है the abundant supply of blood vessels. So these are the structural features जिसके वजह से absorption हो सकता है, होता है majorly in the small intestine. आया समझ में? Are you clear about the answer that they have given in the model sheet? आया समझ में?
Something about lacteals.
No, lacteals are the lymph vessels which are present in the villus. Lacteals are the lymph vessels. हाँ बोलो और क्या डाउट है?
The question was actually mentioned 3 structures of the small intestine, which adds to its absorptive capacity. Which one of the 3 structures which adds to its absorptive capacity. So we have to only write about one structure. Because I've written the narrow structure, the long elongated tube, and its coil. And I've also written that the inner surface contains villi, which adds to the villi adds to its absorptive capacity. I've got full marks.
So यही तो मैंने डिस्कस किया.
I thought we had to write about the why is it narrow and why does it being narrow helps in digestion.
Question ये है कि if I translate in Hindi, question ये पूछा जा रहा है कि special feature small intestine में क्या है जिसकी वजह से absorption हो सकता है? It's a question. So what am I supposed to write? I am supposed to write the extensively coiled nature. इसकी वजह से absorption होता है efficiently. And about the villi, and about the blood supply and the lacteals, we can write very good. Surface area increase हो रहा है. So all these things are the additional points. No. Thin epithelium. So total 5 features है. उसमें से 3 जो क्या है? Did you write all the answers? Yes.
Nutrition.
Did you answer all the questions? When I supposed to get the paper?
Definition.
When I supposed to get the paper?
I have not got it.
You got already? The test was of 15. Yes. How many of you got more than 14? 14 or more, I mean to say.
I don't know 14
I have more 14 or more, I said. Very good. बाकी लोगों को क्या problem हो गया? What happened? What happened? 14 and more, I should say, excellent it is. But the rest, where was your doubt? Because in this 15-mark test and the questions I saw, the questions were elementary questions. Basic questions this was. So, इसमें क्या problem हुआ? हाँ, क्या problem? Those of you could not make, how many of you got less than 10? Not for embarrassing anybody, but I as a teacher have to find out किसको क्या problem हो रहा है. And you have to take it a very sporting way. Next test में ऐसा mark मुझे बिल्कुल नहीं चाहिए. Yes, I'll break every bone that you have in your body now. I'm talking about your test performance and तुमको अच्छा मिला खराब मिला, you have to reciprocate and you have to have that sense in you कि अगला test में मुझे अच्छा कर. That is why your parents are sending you, not to say good morning and good afternoon to us and then go back home. How much will you get? Did you get your paper?
Yes.
अरे
Yellow.
Yellow?
11.
उसका दिमाग कहीं और है. वो थोड़ी ना बायो क्लास में भी है. 11 you got? Still good. I hope nobody got below 10. क्यों? तुम लोग बस काफी बाद में तुम लोगों ने ज्वाइन किया. Did not cover up the portion. अच्छी तरह से पढ़ नहीं पाए पूरा. अच्छा ठीक है. What about you? Below 10. अच्छा make it up. हाँ next class next test में make up करना. What about you? Below 10. Please. जिन लोगों को below 10 मिला है, not to remember all of you have the potential to get 14, 15 ये वाले test because the test was very simple. Some of you had put in your effort, so you got. Some of you did not put in your effort. Some of you joined late, so that was also one reason. So next test में cover up करो, ठीक है? And वो previous chapters पढ़ते रहो, जो भी doubts है previous topics में doubt है, clarify करो in the class. बोलो.
The thing is, epithelial layer is a structural feature of the small intestine as a whole or villi?
But in epithelium of the villus, you can also write if it is asked what is the special feature of the small. That also you can write. Because the fine thin layer of the villus is also adding to the absorptive capacity. I said, इसमें 5 points हम generally लिखते हैं. 5 points are there. You are asked to write only 3. Villus का भी ये special property तो है ही, but अगर small intestine को पूछा गया then that also you can write. Now, what are we left with this part of the chapter is the last part of the chapter, that is the process of filtration of the blood. So that is what we are left with this. The last class, you had already drawn the diagram of the nephron. You had already drawn the diagram of the nephron. What are we going to learn today is, nobody will talk.
Ma'am, I want to say one thing. Not regarding studies, but I want to say one thing. Ma'am, the thing is, you told that anyone can do, or anyone who gets less mark can score higher. I'll tell you one thing, ma'am. In our batch, this batch, there are some students which, if someone scores better than the last exam, then that student does not, I don't know what to say, but that student is not.
Open up. Not to worry. I'm with you. Follow.
That student thinks that no one can grow so high, no one can jump so fast in any exam. If someone scores very less, no one can do good and cannot score good marks.
कोई बात नहीं. इधर आ. What's your name? What is your name? Priyansh. I scared that 5 minutes. No talking. I see anybody talking here, I'll break your bones now.
And also there are some students who talk every time, and then they,
They feel that they are the superior.
yes, and no one can cross them. And if someone crosses them, they are like, she or he did it something by
Cheating.
or cannot grow that fast.
Now you tell me, Priyanshi. That person is telling me, I cannot grow. Does that matter to me? No. That person is telling me that I have got the marks by cheating, where I know that I have not got it. Does that matter to me? Should that matter to me? No. Let that person do whatever that person does. You are a beautiful girl with lots of potential I see in you. Don't be bothered about what people say around you. Don't be bothered about what people say about you. I can tell you about myself. I'm just not even 5 feet, but wherever I stand, I make my presence known. Have that much of confidence and personality. Your parents are there with you to support, your teachers are there with you to support. Even if there is an issue like this, उनको अच्छा मार्क्स मिल रहा है नहीं मिल रहा है, that your teachers and your parents will see. X, Y, Z around you, not to bother. If somebody is boosting you up, you understand, okay, that person is boosting up. If person is there, if somebody is there who is not boosting it up, if they are not encouraging you anyway, ignore and go ahead. Ignore and go ahead. Because if they are getting 90%, उससे भी तुमको कुछ फर्क नहीं पड़ने वाला. If they are getting 20%, उससे भी तुमको कुछ फर्क नहीं पड़ने वाला. Your life is based on how hard you work there. And your parents is support. That's it, no. तो उससे कुछ भी फर्क नहीं पड़ता है. Now here or in school. Be that smart. The same thing I tell my daughter also. Be that smart and confident in. Have that confidence in yourself. I can do it. My teachers are with me. My parents are with me. This X, Y, Z क्या बोल रहा है नहीं बोल रहा है उससे कुछ भी फर्क नहीं पड़ता है. And who that X and Y is, today he is getting 20 on 20 or 80 on 80. You never know. We have seen students 95% वाले आराम से 70 में गिर जाता है. We have seen that. Life में कब किसका क्या होता है and the turns of the life you never know. So कुछ भी फर्क मत, don't take it on you. कुछ भी फर्क नहीं पड़ रहा है. Who is saying what? अपने आप को हमेशा focused रखो. Be very confident about yourself. आया समझ में? Always your confidence should be at the highest. Before your school starts, please get a better haircut. हाँ?
सर बोलते भी है.
सर बोलते कौन से सर?
मेरे को नहीं बोलते.
Otherwise, your teachers will start tying your hair now. So now, the last topic that we are going to study now is the process of filtration of blood. Let us study the 3 steps. Filtration of blood. To study this, what are the 3 steps that we are going to understand? The filtration of blood and formation of the urine. That is what we are going to study. What are the 3 steps? The 3 steps are ultrafiltration. You have to understand the definition and the process. Ultrafiltration. Second क्या है? Selective reabsorption. Selective reabsorption. Third क्या है? Tubular secretion. These 3 processes that we are going to study now. No talking. 3 processes we are going to study which ultimately results in the formation of the urine after the process of filtration of the blood. So how did we draw the diagram of the nephron? We had drawn the diagram of the nephron. What were the main parts we had drawn? The 3 main parts that we have drawn. Let me just draw a simple structure so that we can. Okay. And then what do we have? We have the blood vessel. Which is the blood vessel we have drawn? Which was the blood vessel? What is this blood vessel known as? The renal artery. First of all, we have got the renal artery. Okay, we draw the renal artery bit. Thicker. And then the renal artery divides into a large number of blood capillaries. What is this network of blood capillaries called? Glomerular capillaries. Then from these capillaries, a network of or a fine narrow vessel will emerge out, and this will again start the process of division to form a network. So this was the diagram we had drawn. Now the diagram that I am drawing now would be actually a comparison. I'll show you this way. No confusion at all. We had actually drawn the diagram this way, that this was the PCT and then this was the loop of Hanley, and then the loop of Hanley goes up again, and then the DCT, no? And then we draw the connecting gap. So this was the diagram that we had drawn. Abhi blood vessels, how did we draw? I told you that draw single line blood vessels this way, and then a network of blood vessels come this way. Blood capillaries, which will ultimately unite to form a broader vein. And what is this? What is the name of this? Renal vein. So here we are talking about parts like renal artery. What is the name of this? Afferent arteriole. What is the name of this? Efferent arteriole. And what is the name of this one? What is the name of this network of blood vessels? I say blood capillaries. That is what only I have written in the diagram there. And then we talk about 5. This is renal vein. So now I start with the renal artery this side and then the renal vein. To understand the process, we are trying to come across after the blood has entered into the afferent arteriole. Now what? After the blood has entered into the afferent arteriole, the blood is flowing in a network of capillaries. The blood is moving by the way of the afferent arteriole, has come into this network of capillaries. And in these capillaries, the property of the capillaries remain exactly the same. What are some of the properties of the capillaries we have learned with transportation? In epithelium, and the capillaries also have got minor pores in them. So ये दो features याद रखना है. So what I'm saying is that अब जब ये, now that the blood is flowing in these capillaries, the blood is flowing at a very high pressure. The blood is flowing at a very high pressure. Why? Because if I talk about these two vessels, one by the way with which the blood enters, and the other by the way which the blood is going away. So if I'm talking about these two vessels, what are the names? Afferent and efferent. Afferent arteriole and efferent arteriole. Are the diameters the same? No. Afferent arteriole is much wider. So obviously the afferent arteriole is bringing more blood. But whatever amount of blood is entering here, can the same amount of blood move by this pathway, by this vessel? No, because this vessel is narrower. So the blood which is now every time being poured here in this glomerular capillaries starts developing a very high hydrostatic pressure. They start developing a very high pressure. So now, if I draw only this part, to understand that what exactly is happening comparatively in the Bowman's capsule and the tubular part, and we are talking about the glomerular capillaries, only a single glomerular capillary that I tried to show you. What are the features of the glomerular capillary? I told you they have small pores and the wall is very thin. The blood is flowing. I already told you the blood is flowing at a very high pressure. Now, as the blood is flowing at a very high pressure, what should happen? The liquid part of the blood easily starts leaking out through the pores. What is the liquid part of the blood known as? Plasma. So that is what is happening here. That now the liquid part of the blood now starts draining out. Now the liquid part of the blood starts leaking out through the small pores, and yes, a large amount of ions can undergo the process of diffusion because the wall is very thin. So यहाँ पे दो चीज parallel हो रहा है. What is this? Number one is because of the high pressure of the blood in the glomerular capillaries, quite a large number of the liquid part, a large amount of the liquid part of the blood is constantly thrashing the walls. Pressure of the blood is very high. And इसी की वजह से what's happening? A large amount of the liquid content is leaking out. And addition to that, the process of diffusion is also happening. The process of diffusion is also happening because the walls are thin. Ultimately, what's happening is a small amount of liquid is gathering on the inner wall of the Bowman's capsule. Bowman's capsule is the capsule structure.
Not all the capillaries, they do not completely occupy the whole.
No, there is a gap. The Bowman's capsule is just like as if a knot of capillaries tightly, knots of capillaries tightly bound, and then the blood is flowing. But between the knot of the glomerular capillaries and the wall of the Bowman's capsule, there is a gap. And the inner region of the Bowman's capsule, again, let me tell you this, that the inner region of the Bowman's capsule is not solid. It is having a cavity inside. It is having a cavity inside. Something like, I'm talking about a ball. And if you punch inside, what's happening? There's an inner cavity formed and an outer structure even, and the inside is hollow. It is something that way. So this network of blood vessels, and then one layer, and then inner space is empty. And then the outer layer that we are talking about. So now what's happening? The blood is flowing at a very high pressure, and two things happening. What I said? Yes, by the pores, leaking of the liquid, and ionic diffusion of the ions and salts. As a result, a small amount of liquid is gathering here on the inner wall of the Bowman's capsule. What is this now called? This is known as glomerular filtrate. This is called glomerular filtrate. Why is it called filtrate? Because this liquid is formed after the process of filtration at a very high pressure, जिसको ultrafiltration बोलते हैं. What is ultrafiltration? Filtration of the blood at a very high pressure, which is happening in the glomerular capillaries. Filtration of the blood at a very high pressure in the glomerular capillaries. Now the question is, why the high pressure? Because the uneven diameter of the afferent and efferent. If the afferent and efferent would have been of the same diameter, तभी ये pressure बनता ही नहीं था. So ये ऐसा है कि जैसे the entry point is big enough, wide enough, and the exit point is narrow. So obviously inside there will be a crowd formed. If your entry gate of the school is big and spacious, but the exit gate is narrow, then what will happen? A lot of students will gather. No? So that is what is happening. The entry point is big enough, wider, broader, but exit is narrow. So whatever amount of blood is entering, that much cannot come out. Whatever amount of blood is entering, that much cannot come out. So that is creating a lot of pressure in the glomerular capillaries. As a result of the pressure, now these are the two things happening here, and the fluid is getting collected kahan pe? On the inner wall of the Bowman's capsule, which is a cup-shaped structure, and this is known as glomerular filtrate. Okay, what is the composition of this glomerular filtrate? What are the substances which have actually come out? A lot of water, enough of sodium ions, enough of glucose, and small amount of urea, small amount of uric acid, and certain different type of proteins, but proteins very less, something like this. So we are talking about the fluid which has been filtered is not actually based on only the toxic substances of the body, but काफी सारी substances which are here are all substances necessary for the body. These are all substances necessary for the body. Then why did they come out? Because the molecules could either move through the pore sides of the capillaries or could undergo the process of diffusion. So are we exactly saying here that ultrafiltration is actually removing out toxic substances? No, we cannot say that, because by the process of ultrafiltration, only molecules which can undergo diffusion, they will come out, or molecules which can fit the size of the pore will come out. What is not coming out? RBCs.
In the slide itself,
very less, very less. What is not coming out? RBCs and a large number of proteins, because they get stuck. RBCs and a large number of proteins which have got a bigger molecular size, they are not coming out. And if the concentration is less here, if the concentration is comparatively less here, then they are not coming out. So which are the substances majorly coming out after the process of ultrafiltration are this. But now I have to understand that if all these substances are lost from the body, is it good? No, we will suffer from massive dehydration. Our body will suffer from massive dehydration. Now, how much of the glomerular filtrate is formed in a day? Something around 160 to 180 liters per day. Per day, in 24 hours time, this much of the glomerular filtrate is formed every day. But the question is that how is it possible? We have got 6 liters of blood only. But that 6 liters of blood undergoes the process of filtration so many times. That 6 liters of blood that we are talking about, yes, every minute a small amount of blood is passing through the kidney. Every minute a small amount of blood. That means a single amount of blood, I talk about only 1 liter of that blood, Out of the 6 liters, only 1 liter of the blood is passing through the kidney almost about 80 to 90 times per day. So that is the reason we are talking about such a big amount of the glomerular filtrate formed.
So what percentage is the water in 160?
Water is almost about 60%. So much.
We have to drink continuously water. We have to drink 60% of 160.
We don't drink that much, no? How much of the water are we supposed to drink? Maximum 2 liters, 2.5 liters.
How does the body replace the water?
वही तो अभी second step में हम यही सीखने वाले हैं. How does the see our body is made up? So now I come to this point.
Also, the blood coming up from the efferent artery does not contain plasma.
No, the blood which is coming out from the efferent arteriole, the blood which is coming out from the efferent arteriole is comparatively thicker. The blood which is coming out by the efferent arteriole, please try to understand. अगर समझोगे नहीं तो कुछ भी, if you are not listening me step by step, कुछ नहीं समझ में आएगा. So what I'm saying, the blood which is coming this way has got a lot of water, lot of sodium, lot of glucose, lot of urea, etc., etc. अभी यहाँ पे the process of ultrafiltration has happened and so many substances have come out. Now that the blood which is moving out by the efferent arteriole is comparatively thick. क्यों? Because so much of water has already come out. Sodium, glucose, काफी सारा essential substances has come out. Is this urine? No, this is not urine because there are so many essential substances left in it. Otherwise, इसको urine बोल सकते थे. This is not urine because if it is urine, it should have a higher concentration of urea, uric acid, creatinine, potassium, and less of water, sodium, glucose. So, ये हो गया first step. What is actually happening in the nephron? A process known as ultrafiltration. After the process of ultrafiltration, now what? The glomerular filtrate gathering in the inner wall of the Bowman's capsule. But as I said, the inner wall of the Bowman's capsule is porous. So this liquid, will it stay here for a long time? No, it will start seeping inside. It will start seeping inside. And as it starts seeping inside, the composition of this fluid keeps on changing. How? Because the composition will change because I'm trying to find out that this tubule has got the blood capillaries very close. And a lot of exchange of substances is what is going to happen between the tubule and the nearby blood capillary. A lot of exchange of substances is going to happen between the tubule and the blood capillary. That is what we are going to study under these two steps: selective reabsorption and tubular secretion. First concept सीखेंगे. So, एक minute. Now, what I'm trying to tell you is, let us try to show a blood capillary. What am I trying to show you? A blood capillary जो tubular part of the nephron के आसपास है, and how exactly the process of exchange of substances are going on. So I am dividing ये जो पूरा tubule that I have drawn, I am just dividing this entire tubular part into 3 regions. First हो गया PCT, second हो गया loop of Handley, and the third region हो गया DCT. You know now what are these, no? PCT मतलब first region of the tubule, that is this region I'm talking about. Loop of Handley means this region I'm talking about. And DCT is which region? You have drawn the diagram already. This is the DCT, the distal part of the tubule, and this is the PCT, the proximal part of the tubule. We are now trying to understand that as the fluid is passing through this tubular region, a lot of exchange of substances is happening with the nearby blood vessel. And इसी के reason से और इसी की वजह से the composition of the fluid is changing. So now I try to make it simple, showing you this way, that now that the filtrate is coming down, it has got a lot of water, it has got sodium, it has got glucose, a lot of sodium, a lot of glucose. Urea, how much? Very, very less. Uric acid, how much? Very, very less to understand. What I'm saying, when I try to compare the first part of the tubule and the nearby blood vessel, मतलब यहाँ पे क्या हो रहा है? That is my first concern, and I try to understand that when the filtrate is moving through the PCT, a lot of reabsorption of the water and the sodium and the glucose is happening. What I said, a lot of reabsorption. What is the meaning of reabsorption? Movement of the ions from the tubular part of the nephron to the nearby blood vessel. So now that the blood which was moving this way, was as it is thicker. No, the blood is as it is thick. I said, ये जो capillary, this is a branch of these vessel only, no? So the blood which is flowing in this capillary, which is comparatively thicker, has got very low amount of water, very low amount of sodium, very low amount of glucose, but a high concentration of urea. So in this blood, yes, urea is too much, uric acid is too much. So this is what we are trying to understand. But sodium, glucose. So now what's happening? In the first region, PCT and the nearby blood vessel, when you compare, you try to come across that the sodium, yes, a lot of sodium has come in, a lot of glucose has come in, and a lot of water has also come in. This is what we are talking about as selective reabsorption. First of all, try to understand what am I saying? I'm saying that a lot of glucose which was there in the tubular part of the nephron, which we had actually named as glomerular filtrate, has started moving towards the surrounding blood vessel, and this is what we are talking about as selective reabsorption.
Why does the
Now, reabsorption, what is the meaning of that word reabsorption? Simple diffusion. So यहाँ पे glucose and sodium and water, why are these moving? Why not urea, uric acid? Are they moving? No.
the concentration there is
Because
less?
यहाँ पे concentration कम है, nearby blood में concentration ज़्यादा है. So this is the basic rule of diffusion that we are studying. And on top of that, adequate amount of sodium, glucose, water I said is moving this side. इसके दो reasons है. Number one, high concentration here. And second reason is ये जो membranes है, membranes of the blood vessel and membranes of the PCT, they are permeable. So what are the two reasons which are enabling the process of reabsorption? Number one, permeability of the membranes. Permeability मतलब movement को allow भी तो करना चाहिए ना. खाली concentration differences होगा and the membranes do not allow the movement, तो diffusion नहीं हो पाएगा. So here what's happening? Though the concentration of these molecules like water, sodium, glucose is higher, it should allow diffusion. हाँ, diffusion हो रहा है. Also because the membranes are permeable. Am I clear till here? बोलो.
Since the concentration of urea is higher in the blood capillaries, so does it move the other way?
Very few. एक आध molecule, दो molecule अगर move होता है by pressure तो होता है, but not passive. Easy transport नहीं हो रहा है because the membranes are not permeable to urea, uric acid.
The urea in the blood capillaries there will not travel from the blood capillaries to
At this level they will not travel, but they will travel down here because the DCT is permeable to them.
What will happen if the urea and uric acid is higher?
यहाँ पे then it's a diseased condition. ये normally तो नहीं होना चाहिए.
Also, if the urea and uric acid leaks into the extra space, it leaks into the space between them all the extra.
Vascular, that's a major structural defect that the body has. Normally ये कभी नहीं होना चाहिए.
Since bird will not be able to survive.
No, some people do have structural defects, so that's a major problem. No, that's a major problem. हाँ, आ रहा है समझ में? Raima, any doubt? Anything? So हाँ बोलो.
Sodium, glucose, and water.
हाँ.
Like
The PCT membrane is only permeable to water, glucose, sodium. That is the reason. The blood capillary also. अभी यहाँ पे we are more to understand that the PCT and glucophanty and DCT का permeability what it is. So I said, what are the two reasons of the process of reabsorption from here to here? Number one, high concentration of water and sodium and glucose. And what is the second point I told you? Permeability of the PCT. So permeability of the PCT also will matter. Yes or no? So permeability of the PCT. So these are the two factors which is allowing the process of diffusion of water, sodium, glucose from PCT to the nearby blood vessel. So now, by the time the fluid is moving down to the loop of Henle, now let us see here what is happening. By the time now the fluid has moved down to the loop of Henle, what is left? A small amount of water is there, still small amount of water is there. What about the sodium and glucose? Comparatively come here. And then some potassium ions also they may be there. So right now, when I talk about that, the fluid has moved down till the loop of Henle. What is the next process happening? By the time the fluid has moved down to the loop of Henle, adequate amount of reabsorption of the water is still continuing. Reabsorption of water is still continuing. So I can summarize this way, that whenever I am talking about PCT and loop of Henle majorly, what type of reabsorption is happening here? Majorly reabsorption of water, reabsorption of sodium, reabsorption of glucose. So these three things majorly gets moved from the tubule of the nephron to the nearby blood vessel. And in one of the chapters later, you are also going to understand the role of hormones. Now, there are specific hormones which actually regulate these processes. Special chemical substances released by glands, which is actually bringing about the process of reabsorption. अभी यहाँ पे ये भी सीखना है कि if a person does not have those hormones, then what will happen? The person will release so much of urine. A condition that we can also talk about is diabetes that a person suffers from. डायबिटीज वैसे तो दो तरह के होते हैं, डायबिटीज मेलिटस जो इंसुलिन डेफिशिएंसी की वजह से होता है, एंड डायबिटीज मेलिटस is also another condition where the person releases huge amount of urine, almost about 4 to 5 liters a day, which is abnormal.
So they have to put on a dialysis.
Only dialysis is not necessarily extensive medication. So now when we are talking about why this reabsorption, our body is designed in such a way that out of this, how much has to get back to the blood? Almost about 95% of the water, salt, sodium has to go back into the blood. And how much can a healthy individual release out in the form of urine? Not more than 1 or 1.5 liters per day. So can we think about the difference? I'm saying glomerular filtrate 160 to 180 liters भी बन सकता है. And under normal condition, a healthy individual should not release more than 1.5, maximum 2 liters of urine. उससे ज़्यादा होना ही नहीं चाहिए. If it is, it's a disease condition.
So because most of the water is getting reabsorbed.
Reabsorbed. Abhi, because the fluid is running down the tubular part of the nephron, maximum of the water, salt, glucose, all is given back to the blood. And that is how the blood maintains its composition. That is how the blood maintains its fluidity. Then the blood thick ho jayega, na to the process of circulation will stop. So every minute when the process of ultrafiltration is happening and all these processes, in a very short time all this happen and so efficiently it happens. Why is reabsorption necessary then? Reabsorption is necessary to maintain the fluidity of the blood, composition of the blood, to maintain the temperature, blood temperature, the body temperature, so all these. So come down now as the how does it affect the body temperature? Because if the process of circulation slows down, the amount of energy in the form of heat released by the cells also will come down. We have already studied the concept of cellular respiration where the energy is released and energy maximally is released by ATP and a small amount is released in the form of heat also. No. So now that the fluid comes down, which we now say as the forming urine, because the amount of water is becoming lesser, amount of glucose is becoming lesser. So more and more water going this side, more and more glucose going this side, more and more water, more and more water. And now the fluid which comes down in the DCT will suffer from some major changes. That is, ये जो potassium ions and then uric acid and then urea and then creatinine, whatever was there in the blood which could not move this way so far, all these substances which were in the blood but could not move this side, process of diffusion could not happen. Though they were in a higher concentration in the blood, but the DCT membrane now can allow their transfer I said these type of substances can now move here, but could not move here because of the permeability differences.
Did we say that the function of PCT and this glucophenolate is almost the same?
No, PCT and lupofenil majorly does the reabsorption of water. And PCT does the reabsorption of maximum amounts of water also, glucose also, sodium also. So I would say that the PCT is permeable to sodium, glucose, water. Lupofenil is permeable to water, majorly to water. And what about the DCT? Now that I'm talking about that, this is the DCT membrane. Does the DCT membrane allow movement of water, sodium, glucose? Comparatively lesser, but it is now allowing the absorption. It is now taking. It is allowing the inward movement of what? Of potassium, uric acid, urea, etc., 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 etc. All the toxic substances which are formed or remaining in the blood so far, and they can now move this side. What is this process? Yes, this is also diffusion because the ions and the chemical substances, the nitrogenous waste. Which are the nitrogenous waste here? Urea, uric acid, other major toxic substances which are present in the blood, and they will be moving now this side. Process is same. But if you ask me that why the urea, uric acid could not move this way, why the urea, uric acid which was there in the blood here also could not move this way, my answer straight away will be because the membranes of the PCT and the lupofenil does not allow their transfer, meaning they are not permeable to these ions. The PCT and the lupofenil is not permeable to urea, uric acid, potassium, creatinine, etc. But they are only permeable to water, sodium, glucose. Major. Am I clear?
So what happens to proteins?
Proteins also, little bit of the diffusion will move this side, and some additional proteins will again come back here.
But ma'am, the balance between the potassium and the sodium, the ions on each side should be maintained.
Should
Some
be
sodium will leak into the
If I talk about the DCT, it's small because by the time the fluid has run down till the lupofenil, major amount of the sodium has already gone back. Up. Will it remain in the urine? Should sodium remain in the urine in traces? In traces it should remain or does remain. But zyada concentration if it is remaining, then again it's an abnormal condition.
So most of the potassium in the blood will diffuse to the
potassium excess, whatever it is, will come back here. If the potassium is there in the blood, excess, whatever amount of excess, once the amount of potassium here and here is balanced, potassium transfer will stop. But potassium in excess amount, whatever is there, will start diffusing. Soluble vitamins, urea, uric acid, creatinine, some important ones only I have written here, they will start diffusing. So ultimately now, the fluid which is moving from the DCT to the collecting duct, this is what we are talking about as urine. Now let us try to find out that what is actually the concentration of the fluid which is running down in the collecting duct and what do we call it as? This is urine that we are talking about. And what was this process? This process was known as tubular secretion. Then what is the difference between the word or the process? Reabsorption and secretion. If I talk about the diffusion of substances from the tubule of the kidney to the blood vessel, that is reabsorption. And if I talk about the diffusion of substances from the blood to the tubule of the kidney, then I am talking about secretion. Am I clear?
Yes.
Am I clear? What is tubular reabsorption? The diffusion of substances from the tubular part of the nephron to the blood vessel. Reabsorption. Reabsorption क्यों? Substances should come back to the blood. So reabsorb करना, फिर से लौट के आना. That is called reabsorption. तो substances, essential substances जो tubule में आ चुका था, वो तो lose नहीं होना चाहिए. So the body is made up in such a way that it should get back into the blood. It should come back into the blood to maintain temperature and the fluidity and the blood pressure, everything. Otherwise, within few minutes, we may collapse. अगर ये process efficiently नहीं होगा. Then what? अच्छा, so I said during the process of or in the PCT and the lupofenil, enough of reabsorption. DCT में the process is just the reverse. DCT में reabsorption नहीं होता, but secretion happens. Now what is secretion? Secretion, the basic meaning of secretion मतलब release करना. Release करना. So now I'm saying secretion. Why the word secretion is being used here? Because the blood is giving away all the toxic and unwanted substances to the fluid in the tubule. The blood is giving away all the toxic and unwanted substances to the fluid in the tubule, which is going to come now into the collecting duct. And this is what we are going to term as urine Q, because it has got only a small amount of water, which is necessary to dissolve the toxic substances like urea, uric acid, any other type of substances. Now when we talk about the word drugs and poisons, we take food along with preservatives, we eat medicines, we take so many other different type of substances. So that is what we are talking about as drugs and poisons. Actually, additional amount of potassium ions, so many different type of soluble vitamins. So ये जो composition है of the fluid now which is running in the collecting duct, and then it will enter into the urinary bladder, no? It will go into the urinary bladder. From the bladder, where should it go? Into the urethra. From the bladder, it should go into the urethra. So this is the fluid that we talk about as urine. What are some of the properties? This is concentrated. This is not dilute. This is concentrated and light yellowish in color. But
urine is 95% water. How is it dilute?
Urine is? 95%. No, 95% water, and the amount of salts, comparatively when you talk about it, so much of salts that it is dissolving with only a small amount of the water. We are not talking about water and hardly any amount of salts and ions. So much of salts and ions comparatively. Comparatively, we are talking about. So the urine that we talk about is actually concentrated, but a light yellow tinge it has because of a particular pigment formed in the liver. Now you get questions that what are the main nitrogenous waste substances? Urea, uric acid. Where is urea, uric acid formed? Is it getting formed in the liver or in the kidney? It is getting formed in the liver, and by the way of blood it is coming here. Remember, the kidney is only doing the process of filtration. Kidney is not making anything, no chemical substances it makes on its own. Kidney का कोई secretory function नहीं है. It cannot make any type of chemical substances. Where are these chemical substances known as urea, uric acid formed? Liver. Amino acids. By the way of excess amino acids which gets deposited in the liver, the amino acids can get converted into some other type of chemical substances with urea as a byproduct. Okay. So let us write some one or two questions here. Ultrafiltration. Let us try to understand how to define these words. Define ultrafiltration. Ultrafiltration. How you should define this word? Any doubt in this? Please ask questions. Ultrafiltration. The filtration of blood. The filtration of blood. The filtration of blood in the glomerular capillaries at a very high pressure. Filtration of blood in the glomerular capillaries at a very high pressure. Filtration of blood in the glomerular capillaries at a very high pressure. Okay? Now next is selective reabsorption. You are asked this definitions for 1 mark. After that, I'll give you some more questions for which the concept will be clear.
Can you please repeat the definition again?
Ultrafiltration, the filtration of blood in the glomerular capillaries at a very high pressure. In the glomerular capillaries at a very high pressure. Next, selective reabsorption. Write down two differences between selective reabsorption and tubular secretion. Make a column. Make a column. There only I'll give it. There only I'll give it. There only I'll give it. They're running out of time. Make a column. Let us write two differences between selective reabsorption and tubular secretion. अरे यहाँ पे लिखो ना. She has her parents to look after her and think about her. You please don't take that pain.
Selective reabsorption.
Selective reabsorption and tubular secretion. There only I'll give you the definition also and some other differences. फटाफट लिखो. Selective reabsorption, tubular secretion. ठीक है? देखो first point, the definition itself, the diffusion of तो माता से ट्रैम्पलिंग यूज सो मच नो इसलिए वो स्केल और पेंसिल निकालने में तुमको आधा घंटा लगता है ना चलो selective reabsorption, the process of diffusion or the diffusion of essential substances. Diffusion of essential substances. The diffusion of essential substances from the tubular part of the nephron. From the tubular part of the nephron to the blood vessels. Tubular part of the nephron to the blood vessels. Other side, tubular secretion. The diffusion of toxic substances. Diffusion of toxic substances. Diffusion of toxic substances. Once you understand, you can write. Remember what you are writing. Diffusion of toxic substances from the blood vessels. From the blood vessels to the tubular part of the nephron. From the blood vessels to the tubular part of the nephron.
Can we write the plots?
You can write that, that's very good. But these terms not there in the book, so I'm not stressing so much, not making it compulsory. If you can remember, very good. Chalo, second point likha, first one ho gaya. Second, the diffusion of harmful toxic substances. I said diffusion of toxic substances from the blood vessel to the tubular part of the nephron. Blood vessel to the tubular part of the nephron. What are these? Not blood capillaries are also blood vessels. Capillaries भी तो blood vessels हैं. Blood capillaries भी तुम लिख सकते हो. That's not a mistake. चलो number two. Second difference. Under reabsorption. Substances such as water, sodium, glucose are reabsorbed. Substances such as water, sodium, glucose, proteins also can add there. Water, sodium, glucose, proteins, etc. Water, sodium, glucose, proteins, etc. are reabsorbed. Secretion will go. Substances such as urea, uric acid. Substances such as urea, uric acid. Excess potassium ions, soluble vitamins, etc. are secreted. I'll say again. Substances such as urea, uric acid, excess potassium ions, soluble vitamins, etc. are secreted. Turn to your volume. This you should complete as homework. Turn to your volume. This you will complete as homework. Now the question that I ask you, just 2 more minutes. All of you take out your volume and turn to page number. Take out your volume, turn to page number 177. Turn to page number 177. अच्छा dialysis that is left. Dialysis will talk about in the next class.
177.
हाँ 177. Now फटाफट pick some questions. 17. I'm sorry. 177. Question number 50. Question number 50. Question number 50, 51. 177. 50, 51. 50, 51. Come to 179. 179. 23. 23. 23. 29B. 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 182. 182. 59. 182. 59. The next class I'm going to start with the next chapter, control coordination. But the last एक छोटा सा topic बाकी है, that is excipients, glands and dialysis. Dialysis also there. That we will take 10 minutes to discuss and I'll move on to the next chapter. So this question all of you should complete.
पर अगर मेरे गांव में
दो मिनट दे दो मिनट
अरे जाएगा